So mutations have two general causes. The first cause is just that life causes mutations. These are spontaneous mutations. They just happen because of abnormalities that occur during normal biological processes. So this is things like mistakes in replication. They also can happen due to byproducts of metabolism. So basically, just the price of being alive is that sometimes you get mistakes in your DNA. The rates can vary from species to species, so some organisms will have a very rapid rate of mutation, and other organisms will have a very slow rate of mutation. Even within the same organism, some genes can be more prone to mutations. So certain genes in humans, for example, are much more prone to mutations, and so they're more likely to end up with disease. And then other genes are very stable, and they very rarely get mutations. In general, we can say that you should get about 1 mutation for every 1 million genes as kind of the background rate of spontaneous mutations. This is an important number because this is how people figure out if something extra is causing mutations. So if I know that I should only get this many mutations, and I start to see an increase in the number of mutations that an organism is getting, that tells me that there is something in the environment that is causing those mutations. And if there's something in the environment that's causing those mutations, they're called induced mutations. Induced mutations are caused by environmental agents. And again, they're going to cause the mutation rate to be much higher than the regular background or spontaneous rate of mutation. Agents that cause mutations are called mutagens. So that literally means creating mutations. And they can be chemicals, they can be physical things like UV or X-rays that can alter the DNA in various ways. Mutagen may sound like another word you've heard before, carcinogen. A carcinogen overlaps with a mutagen. Carcinogen means to cause cancer. And because cancer starts with mutations, many carcinogens are also mutagens. So there's some overlap in these terms. Something can be a mutagen and a carcinogen. But the reverse is not true. Not all carcinogens are mutagens. Many carcinogens are irritants because the other aspect of cancer is too much growth. So cancers have mutations, but they also grow too much. And so carcinogens, if they don't cause mutations, might just irritate the tissue and cause it to grow too much. And so tobacco is a really potent carcinogen because it does both. It causes mutations and it's an irritant. So you're doing both things. You are changing the DNA and you're irritating the tissue so that it divides more. And that's why it's such a significant carcinogen and why it causes a large amount of lung cancers. As well as, and this is starting to be more and more common, oral and throat cancers. So things like having to have your tongue removed or something like that. And I like to mention that if you're a smoker, you should really consider quitting because it is such a powerful and dangerous combination. And the good news is that if you quit today, your body can fix the damage, and 10 years from the day that you quit, your risk for cancer is back to somebody who never smoked. So sometimes I hear people say, well, I'm just going to keep smoking. I've already done the damage. Well, that's not true. If you quit today, you could undo that damage and be back to the risk of cancer that somebody who never smoked had 10 years from now. So I'd encourage you to think about quitting. So we're going to go through some examples of mutagens. Spontaneous mutations, as I mentioned earlier, are just mistakes in DNA replication or the result of metabolic byproducts and some other things we won't talk about. Induced mutations are going to be chemical mutagens or physical mutagens. There's lots of different chemicals that cause mutations. Benzopyrene is one that's in cigarette smoke, for example, and there's lots of other ones. Physical agents are things like UV and X-rays that can damage the DNA. So you don't need to memorize the different names of some of these chemicals, but I'm just going to show you some examples of how these cause mutations. So this is an example of one nitrous oxide. And so what nitrous oxide does is when you have a normal sequence, so let me write some sequence here. You have a C, A, G, T, and what happens is that the cytosine will be replaced with a uracil. So now, so let me draw my original sequence. Sorry, let me finish doing this. So this is my original DNA. And what happens is that the nitrous oxide, and let me change colors for my mutated DNA, it's going to turn this C into a U. So it literally chemically modifies it into a U. Now, should there be a U in our DNA? No, right? So that's normally only an RNA. So the cell gets very confused and it tries to fix this. So the first thing that it does is try and make the right base pair. So it's going to go ahead and pull this out and put it in A. So now you have an AU base pair, right? And then the next fix is that it's going to take out that U and put in a T. And next thing you know, I have an AT base pair instead of a CG base pair. So there's first the damage that the chemical does, and then there's the process where the cell tries to fix it. And when it fixes it, it ends up with the wrong base pair. Same thing is happening here. This chemically changes the adenine into a base called hypoxanthine, which is not a normal base, but it's a modified base. And hypoxanthine pairs with C. And so now the cell will try and fix this hypoxanthine, and you will end up with a CG base pair when the DNA gets copied. So again, you get the damage, and then you get the repair and copying of this into the wrong base pair. Other chemicals might actually insert into the DNA and bend it. And when you distort the double helix, it messes up the shape of the double helix, and that affects the way that the DNA replication occurs. So then it will cause replication to get messed up, and then you can get substitutions as well. So there's lots and lots of different chemical mutagens. They all work in slightly different ways. These are just some examples. Physical mutagens work depending on their strength in different ways. So remember back to the electromagnetic spectrum, we had shorter wavelengths increasing the amount of energy. So remember there was light here, and then over on this end there was radio waves that have relatively low energy. Then you had UV, you had X-rays, and you had gamma rays. So UV has more energy than light, but has less energy than X-rays. X-rays have more energy than UV, but they have less energy than gamma rays. So because of these differences in energy, they'll damage you in different ways. X-rays and gamma rays come from radioactive materials. And X-rays pass through your body. They have so much energy, so they actually go through you and they pass through your cells. And as they pass through your cells, they can hit the DNA. And they're carrying so much energy that they literally snap the DNA in half. And when it snaps the DNA, it can make ragged edges. So imagine, again, this is my original sequence. So there's the matching strand. And it's just going to crazily snap this DNA into pieces randomly. So now I have a piece that has ATAGC single-stranded, right, and the rest is double-stranded. Then I have like a TA, a different piece that's TA. Whoops, TA is still here. And then I have another piece that has the G, and then it has this little single-stranded area, and then the rest of the DNA. So in these parts and these parts, these will have to get fixed. But because they're messed up and broken, when they go to get fixed, it's really common to get mistakes. So the cell will try and fix this, and then it has to paste this back together. But it's kind of like, you know, if you've ever broken a ceramic bowl, you can glue it back together, but it's usually never quite the same. You might have a crack that doesn't quite seal or a piece that's missing. And so again, when we try and fix this break, our cells sometimes will make a mistake. So that's what happens with gamma and X-rays. And UV rays have less energy. And so because they have less energy, they don't break the DNA. But they have enough energy to do something really unusual that doesn't normally happen in the cell. So this is showing you before. So here's the original original double helix. So there's a T, T, A, A, and then those two dots between them is representing the hydrogen bonds that are normally between them. And that's all that's normally happening with the base. So what happens when UV is exposed to this is it creates this unusual covalent bond between two T's that are next to each other. So remember, bases that are next to each other are not normally bonded to each other. They're only bonded through the sugar phosphate backbone. So the bases themselves are not bonded in the original strand, only the sugar phosphate is bonded. That's the ribbon part here. Ultraviolet light will cause this extra covalent bond, and it's a double bond between the two T's. And this is called a thymine dimer. And you can see that the problem with a thymine dimer is it creates a little bump in the DNA. It distorts the double helix. And so these are going to have to be fixed. And when they get fixed, there can be a mistake that occurs. So again, it's kind of this repeating theme. A mistake happens, the cell tries to fix it, and when it tries to fix it, it doesn't always fix it correctly. You get millions of thymine dimers all day long when you're out in the sun. It's just a part of being human. So our bodies are going to constantly fix thymine dimers. But this is why there is a possible association between UV exposure and skin cancer. So I mentioned already that mutations are a normal part of life, right? So especially certain kinds of mutations. So we can't ever avoid spontaneous mutations. And we can't avoid a certain amount of mutagen exposure. So for example, humans have always lived out in the sun. So UV exposure has always been part of the human experience. So for this reason, we have DNA repair mechanisms that we've evolved to deal with this normal part of human experience. Now, I will mention, however, because I think this is important, that modern humans experience a much greater rate of environmental exposures to mutagens. And the number one reason is, of course, because of human-created chemicals in our environment. And so estimates are that 75% of cancers are directly caused by environmental mutagens that humans are exposed to. And that's a pretty high number, and this is why we probably see much higher rates of cancer in the first world, because we're exposed to much more chemicals. And again, we haven't evolved to deal with this much DNA damage. We evolved to deal with this, not all of these chemicals that are in our environment. So be careful about the environment of the chemicals you expose yourself to. You absorb chemicals in lots of different ways, and people don't realize that, say you put lotion on your skin, all of the chemicals that are in that lotion directly absorb into your bloodstream and are distributed to every cell in your body. So it's what you breathe, it's what you smell, it's what you touch, it's what you eat, can all be providing this exposure to environmental mutagens. So how do we fix our DNA and try and fix these problems that happen due to mutagens? Well, it's really two parts. First, your cell has to be able to tell that the DNA was damaged, and then it's going to have to fix that damage. So two different steps usually. First, I need to know, then I have to repair it. The biggest way that the cell knows that there's damage is wrong base pairs. So remember that there's normally a purine-pyrimidine base pair, so like A to T and G to C. And the purines and pyrimidines have the double ring, and the pyrimidines have a single ring. So as long as I have a purine paired to a pyrimidine, I have 3 rings of base inside of my inside of my DNA. So if I imagine the width of my DNA, this is my double helix, I have my 2 rings and my 1 ring. So the width of my double helix is always 3 rings. If for some reason I have a wrong base pair where I put a purine next to a purine, so this might be an A next to a G, this is not a correct base pair. If I looked at my DNA, it's going to be a bulge because now I have 2 rings base paired with each other instead of the 3 rings that's normal. So that's one of the primary ways that DNA damage is detected. Then once it's detected, it will be repaired, and there's sort of 2 broad ways to repair it. And the first way is to directly repair the mistake that happened. So if some change happened to the DNA, then I would undo it. So for example, so let me draw out an example here. Let's say I had TAT, and that was mutated to, so let me put in my mutation here, a G. The repair process is just going to directly convert that back into TAT. So that would be direct repair. Excision repair is to cut out. So excise means to cut out. So excision repair would look like this. So there's my original sequence. This is mutated to a G. So the repair process would take out the G. So now I just have my T and my T. And then in a second step, it would fill back in there with the A. So it cuts out this part and then fills back in. This is the most common type of repair because it can fix anything. So this is analogous to your delete button, right? You can delete and then fill in with whatever you want. Imagine if you had a keyboard where instead of hitting delete, you had to have 100 buttons. And one button was for delete A, another button was for delete B, another button was for delete C. So for every letter of the alphabet, you'd have to have a different delete button. You'd have to have a huge keyboard, right? And push delete A, and then you could type back in what you wanted to type back in. Delete B, the whole separate button to delete. So for every mistake that you made, you would have to have a different way to fix it. So that's not very efficient. It's more efficient to have the one button, the delete button, that can undo anything. And so again, that's why that's the most common type of repair. So I like to end this chapter because mutations can be kind of scary to people. It can be kind of emotional if you've had anybody in your family who's had cancer, by reminding you that cancer isn't a single mutation. It's an accumulation of lots and lots and lots of genetic changes. So it's many accumulated mutations in the same cell. Most of us, by the time we're in our 20s, already have mutations in lots of our cells. But it's many accumulated mutations in the same cell, combined with an increase in growth. And so when that happens, that's what can lead to cancer. And so many mutations, it's so many mutations that they can actually have whole chromosomes that are different. So this is what a normal human chromosome makeup looks like. So we have 46 different chromosomes, and each one of these, so one here, one here, one here, one here, each one of those represents a double helix that has hundreds and thousands of nucleotides. In cancer, and so this is from a cancerous cell, you don't just have 1 or 2 mutations here and there. You have mutations that are so big, and I'm looking at this section here, you have so many mutations that you actually have chromosomes that are unidentifiable. So this is hundreds and thousands of nucleotides that are different from the original cell. So that's why, cancer is still relatively rare, a relatively rare event, and something that happens much later in life. And so that wraps up mutations.